Hello everyone, my name is Pol del Aguila and I'm a scholarship holder for this year, and I'm here today to thank all of you for your help and your support, especially to those that gave money to the KTH Opportunities Fund. So, I'm a PhD candidate at the School of Electrical Engineering and Computer Science here at KTH, and thanks to this wonderful support, I actually got to travel to very important universities around the world, like Cambridge or Stanford, and very important conferences like ISB in Washington or ICASP in Canada, in Alberta. So let me tell you today a bit about what was the research I did and I went to present there and how well it went. In this case, the research we did was related to biomedicine. In biomedicine, they have some experiments called immunoassays, and these immunoassays are used to establish whether, for example, a prototype of a vaccine is working, or in some cases in hospital care, whether a patient has a specific disease, like for example tuberculosis. Once these experiments have been prepared, one waits for around 24 hours to get any kind of answer from these experiments. During these 24 hours, what happens is the following. Some of the cells in the experiments inside these microwells, cells such as these two here, they generate some important particles for the results of the experiments, and these particles, they follow these trajectories that are random. And some of these particles get stuck to the bottom of these wells that are prepared specially for that, while some others are moving freely. To get an approximate picture of how this looks in reality, we have here this simulation. Here you'll see that there are some green dots that are moving freely, while there are some red particles that are stuck to the bottom of the well. And some of the red become green and move freely, and some of the green become red and get stuck there. At some point, however, the experiment finishes, and the result of this whole thing is an image such as this one. A natural question at this point is to say, well, where does it say whether the vaccine works or whether the patient has tuberculosis? And the clue to this answer actually is inside this image. It depends on the number of cells that have generated these particles and then show up in the final image. Of course, what you're just seeing, it's me trying to pinpoint how many cells there are. And as you can see, there are some debatable decisions. And the intuitive question here is whether there is a more automated, principled way of doing this, right? In other words, why doesn't a computer do it instead of me? Up here, we're going to look at what it's called the cross-section of one of these spots. The cross-section is the brightness as we transverse one of these spots in a line like this, like I've just drawn. So basically, for a computer to be able to find this kind of cell, but also this kind of cell, it has to be able to distinguish, to find both this kind of curve and this kind of curve. And the problem here is that computers like to compare stuff. And for example, if one were to establish that the rule to compare whether there is a cell or not is to compare with this black level here, this cell here would be found, but this cell here would be ignored. While if one were to establish the comparison value down here, one would find both cells, but at the same time, one would be exposed to mistakes, like, for example, this little spike, or this one, or this one, or this one, which would all be detected as cells when, in fact, they're not. This pattern that computational methods actually struggle to say that things as different from one another as this one and this one are actually the same while it's so obvious to a human that this is a spot, while this is also a spot, it persists in much more complicated computational methods. So what we did to improve upon this was to use partial differential equations to try to explain the problem. And don't worry, I'm not gonna get into the details here, but basically we modeled the problem by using three quantities. The C, and I've plotted it here in green, is the density of particles that are floating around, like in the simulation we saw before. The D is the red particles in the simulation, so the density of particles that are stuck to the bottom of these experiments, while the S is the new particles being thrown in. Clearly, if anyone knew S already, we wouldn't have a problem because it contains the number of cells that there are there. Our new way of solving these partial differential equations allowed us to express our image observation in terms of something that is pretty similar to S in that it gives us all these positions. We then use tools from fields such as functional inverse problems or non-smooth convex optimization to obtain an f-1 that could, from an image, give us A and then the position of these cells. All of these new results and the hard work of 2 years gave us an algorithm that can actually do this as well as humans at a comparable quality. And thanks to the support from KTH Opportunities Fund, we were able to go around the world to explain to other researchers our success story. The first stop of this journey was in Cambridge, in the Isaac Newton Institute of Mathematical Sciences. There, not only did I meet very smart people from around the world, but I also presented a presentation and a poster that are both available in my website for download if you're interested. The next trip had several stops and took me to explore North America. First in Washington, then in Palo Alto, California, and then in Calgary, Alberta, Canada. In Washington, I had the luck to attend one of the most important conferences in biomedical imaging in the world. There, I attended a tutorial on optimal transport that was not only great but very useful too, and numerous poster sessions and great plenary talks. Actually, in one of these poster sessions, I presented the work that we've done here at KTH, which is also accepted as a paper publication in the conference proceedings. For my remaining 2 weeks traveling through North America, I would direct you to the KTH alumni Instagram account, where I reported my activities during those days. I hope it doesn't disappoint. It includes the hot, good weather of California, the cold of Canada, an amazing visiting week at Professor Stephen Boyd's lab in Stanford University, Electrical Engineering, another poster presentation, and another accepted proceedings publication. All of this said, I would like to thank you all for organizing and contributing to this fund and wish you a good day.